I'm Dr. Double B, and this is my lecture on solving all of the world's problems with NGS, or next generation sequencing. NGS allows sequencing of much more of the genome than was previously possible, though now it's been around over a decade. There are a few major technologies, namely Illumina, ion torrent, and long-read systems like PacBio and Nanopore. Illumina's process begins with fragmentation of the DNA, either by ultrasonication or an enzyme. Adapters are ligated, which allow the DNA fragments to bind to an oligonucleotide bond. They then undergo isothermal bridge amplification, which amplifies the strands locally so we have enough to detect. In the sequencing step, reversible terminator nucleotides with fluorescent labels are added. And at each cycle, the camera takes pictures of the flow cell before the terminator is cleaved and another nucleotide is added. In the ion torrent procedure, DNA is fragmented and attached to beads, where it undergoes an emulsion PCR reaction, giving us enough clonal DNA to detect. The beads are added to wells, and nucleotides are added, one specific nucleotide at a time. If incorporated, the hydrogen ion release causes a current change which can be detected. There's a known issue with homopolymers here. After about 4, you lose the ability to count how many were incorporated. There are a couple steps that usually get skipped in the animations. Target enrichment, where the regions of interest are selected, which we'll get back to later, and indexing, where oligonucleotide barcodes are added to the samples so they can be multiplexed. In another emerging technology, nanopore sequencing, whole DNA molecules are ratcheted through a nanopore, where current changes are detected and translated into specific nucleotides. I'll be focusing on the first two, more so Illumina, since that's what I've worked with the most. With Illumina, the first file type we get back is the BCL, which contains raw data that still needs demultiplexed and converted to FASTQs. A FASTQ file looks like this. You have some data about the read, the sequence itself, and some gibberish that corresponds to read quality at each base. This is called a Phred score and corresponds to probability of an error. The interesting thing about the FASTQ file is you have no idea where the sequences come from on the genome. So the next bioinformatics step is to align each read to the human reference genome. This uses a combination of hashing to find the region it maps to and local alignment, where the exact best match is found. Of course, you can't always find a perfect match, since the whole point of this is to find variants from the reference genome. Some of the variants it needs to account for are single nucleotide variants, small deletions, and small insertions. Here's a low-power view of the reads stacking up. The The alignment yields a SAM, sequence alignment map, or really a BAM, binary alignment map, compressed version. This file also has the full sequence data, but now it has the chromosomal locations of each read and how well it matched the reference. These files can be viewed directly, usually in IGV, the Integrated Genomics Viewer. You'll see all of the individual reads stacked up against the reference genome. The thin lines at the bottom are the introns, and the blocks are the exons. We usually don't display all of the nucleotides, only the ones that are different from the reference. This is an example of a KRAS G12C mutation caused by a C to A mutation compared to the reference genome, or, and this is a little confusing, a G to T mutation on the cDNA since the reading strand is in the opposite direction. Deletions look like this, with a line displayed where the deletion was. Insertions look like this, with a thin purple line at the position of the insertion, which you can click on to see what it was. One of the important metrics to be aware of is the read depth, how many reads we have at any given nucleotide. Then there's the variant allele frequency, what fraction of reads have a certain variant from the reference. In this example, the read depth is 20 at the highlighted position. There are 20 reads that cover the nucleotide. The variant allele fraction of the A is 35%, since 7 of the 20 nucleotides are A rather than T. In clinical genetics, we want a minimum of around 30x, and in cancer genetics, we want somewhere in the range of 200x. Here's one more animation putting all of these steps together. We have some tumor cells, as well as admixed normal cells, with target cancer genes, one of which has a mutation. The DNA is extracted, and the library prep is performed to select the target regions, which is not really shown here. The DNA hybridizes randomly to the flow cell, where it undergoes amplification. The DNA is sequenced, with each amplified cluster giving us one read. The reads are then aligned to the reference genome, and we see our mutation in 30% of the reads, corresponding to 30% of those DNA strands having the mutation in the original sample. Of course, we don't want to look at 1 million bases or potentially 3 billion bases manually, so the next step is variant calling, which records all variants from the reference. This yields a variant call format, or VCF file, which stores the chromosomal positions and variants. This file can undergo annotation, where we add things like gene name, amino acid change, and population frequencies. Here's a summary of the steps from sequencing to annotation. The VCF data can be viewed in various software platforms where we review variants for biological and clinical significance. The first question is always, is the variant real? We look at things like read depth and variant allele frequency, and may review it in IGV, especially for complex variants. The second question is, does it matter? Most variants are benign, with no major impact on the protein. Population databases can help sort most of these out. If something's very common, we usually assume it's benign. Some variants will be well studied and known to alter the protein function. In genetic diseases, that usually means damaging the protein. In cancer, this could mean damaging a tumor suppressor or overactivating an oncogene. Some variants, unfortunately, are just not well studied and we won't know the effects. In pathology, there's a long history of hedging. For instance, atypical squamous cells of undetermined significance, or ASCUS, and atypical glandular cells of undetermined significance, or AGUS. So we can take a note from them and call ours atypical nucleotide of undetermined significance. So when you're looking in an anus and trying to determine, that sounds kind of bad, doesn't it? Let's call it something else. Let's call it a polymorphic nucleotide of undetermined significance. So when you have a penis and you're trying to determine if it's significant or not, oh wait, no, that's much worse. Let's try variant of undetermined significance, or VUS, VUS, yeah, I think that one's safe. Anyway, Both clinical genetics and cancer genetics use tiering systems to classify variants and deal with this uncertainty. A set of ACMG AMP guidelines focus on germline variants and give a system to try to determine pathogenicity, meaning does it cause disease. Other AMP guidelines focus on cancer, and the tiering is based on clinical significance. Tier 1 variants have a known therapy or diagnostic or prognostic significance, and Tier 2 variants have a potential or experimental utility. There are a lot of approved therapies to keep track of and even more experimental therapies, so we don't memorize them all. This is where databases and guidelines come in. The NCCN guidelines are a major resource for oncologists describing what therapies are options based on various tumor characteristics, such as stage and genomic alterations. These may not have brand new approved drugs in them. OncoKB lists therapies by drug, tumor type, and sometimes specific variants. ClinVar mainly tracks pathogenicity of specific variants. Civic tracks variants and treatments. Nomad reports population frequencies, which can help identify variants as benign. Bioportal and Cosmic have databases of mutations detected across cancer types, though don't report on pathogenicity or significance. Simulation algorithms attempt to predict pathogenicity, mainly by looking at conservation of the sequence where mutation occurs across the genome. People put different amounts of faith in these, and they'll often contradict each other. Now, I've mainly talked about small variants so far, but we also want to catch other things like copy number variations and fusions. Copy number alterations can be detected by looking at relative sequencing depths across different genes. If a region has less coverage than expected, it likely has a deletion. And if it has higher coverage, it likely has an amplification. Here's a case with ERB2 HER2 amplification, where a typical ERB2 region has about 52,000 reads, and a typical KRAS region for comparison has 2,000 reads. Single nucleotide polymorphism data can also be used from NGS results to assess CNVs and copy-neutral loss of heterozygosity. This data can also be used to assess homologous repair deficiency, but I'll leave that for another time. Fusions can be detected by sequencing the region of the fused genes, which is best done on RNA, where you generally only have the fused exons. RNA-seq begins with reverse transcription, converting the RNA to complementary DNA, or cDNA. From this point, it can just be sequenced as DNA. When we look at the alignment of the RNA, it won't have introns, so it's going to have gaps displayed as a thin line, and this is normal. When there's a fusion, part of the read will align to one exon, and the other parts, or the mate pair, will align to another gene entirely. Here's an example of a ROS1 fusion in IgB, where the reads stop abruptly at the intron and the other ends map to another gene. Now, let's rewind a minute. Here's a step that usually gets skipped in the NGS explanations and animations, and that's target enrichment, where you select for the regions of the genome you're actually interested in. This can be done by PCR or hybrid capture. PCR you can figure out for yourself. Hybrid capture uses biotinylated oligonucleotides to capture the regions of interest. Magnetic streptavidin beads bind these complexes, and a magnet keeps the beads at the bottom of the wells while the rest of the DNA you're not interested in is washed away. I bring this up now because it has an advantage in detecting fusions. You only have to target one of the fused partners, so you can capture novel fusions. One limitation is that translocations that don't actually create a chimeric mRNA will be missed, like promoter fusions. PCR-based enrichment methods use allelic imbalance to detect novel fusions when the 5' and 3' expression levels of a gene differ. RNA sequencing also provides additional information when assessing splicing alterations. Here's an example of a MET exon 14 skipping alteration in a lung adenocarcinoma. The top half, which is DNA, shows a deletion of the splice acceptor site. On the bottom half, the majority of the RNA reads show skipping of all of the exon 14. Another application of RNA-seq is gene expression. Like copy number variants in DNA, depth of coverage on RNA-seq reflects how many copies of mRNA were present in the cells. Gene expression can be used for tumor classification and various other purposes. NGS can also detect other biomarkers, microsatellite instability, MSI, and tumor mutational burden, TMB. I won't cover MSI in detail here, but basically, NGS sequences the microsatellites, and insertions and deletions mean the repeat was unstable. TMB refers to how many somatic mutations a tumor has per megabase. The idea being that tumors with high TMB will have more neoantigens expressed on the surface of the cells and will respond better to immune checkpoint inhibitors. The official FDA approval is for tumors with TMB over 10. As an example of a large cancer panel, here's the gene list for the TSO 500. All genes are covered for small variants and many for fusions and copy number alterations. And here are some examples of reports. This case is a rectal adenocarcinoma with ERB2 amplification. This is a cholangiocarcinoma with a targetable IDH1 mutation. Here's a prostatic adenocarcinoma with DNA repair alterations, targetable with PARP inhibitors. One kind of off-label use for this is testing two tumors to determine if they have the same clonal origin, that is, if one is metastasis from the other. Here's a case with lung and ileal tumors where the alterations matched closely, supporting the same origin. Liquid biopsy is another application of NGS, though it can use other techniques too. Tumors shed DNA and RNA in small amounts into the bloodstream. With very sensitive methods, cancer mutations can be detected in the blood without even obtaining tissue from the tumor. With NGS, this requires very high depth of coverage, 20 to 30,000 X, since the circulating tumor DNA would be very rare. There are also a few catches. Tumors may not be shedding DNA into the blood, which is more often true if they're lower stage. You don't necessarily know which tumor a mutation is coming from, and the patient could always have another unknown tumor. And hematopoietic cells can carry background mutations referred to as clonal hematopoiesis of indeterminate potential, or CHIP. The latter can potentially be filtered out by separately sequencing the white blood cells. While I focused on cancer, there are many other applications. Clinical genetics, of course, pharmacogenomics, infectious diseases, and things like ancestry testing and population genetics. One particularly interesting application is sequencing the microbiome. In the colon, for example, this has been shown to be able to predict IBD status. In my lab, we had done some COVID sequencing. Certain variants from the reference define the different subtypes of the virus. So maybe NGS can't solve all of the world's problems, but it can do a lot. Thanks for watching. This is the part where I'm supposed to tell you to subscribe and like and all that, so yeah, do it.